Good afternoon. My name is Vanessa, and I am part of the Human Microbiome Project with Adele and Kariatsu, who are not present right now. So far in Summer 1, what we did was we identified DNA that lives within the human microbiome. Summer 2, we took a different approach. We added a clinical portion with testing antibiotics that are usually found in like hospitals, doctors, and we also did an environmental aspect, which allows us to witness what types of DNA is within soil, so we can identify environmental genes as well. Quick update with the Summer 1 thing is we continued collecting samples. This is another picture of a pierced belly button, and I was very happy. And we did DNA extractions where we literally take a sample, extract the DNA. Then we do quantifications to see how much DNA we actually extracted from said sample. Then we do a PCR, which basically uncoils the double helix, and then from there, we send it to a lab called the GeneWiz, and they give us an ATGC sequence, which allows us to use a specific algorithm that lets us actually know what type of DNA we're dealing with, bacteria we're dealing with. So for the antibiotics, which is the clinical portion, we have a bunch of different samples. We tested samples from 2015 Staph aureus to current day. We used septomycin, ethicillin, cetomycin, and apomycin, and we tested the zone of admission. And as you can see here, these little areas, that's just kind of how strong the bacteria is resilient to them. So then we did another procedure called the SPA. So quick backstory. The Staph aureus is everywhere within us, and it can be categorized based on a protein called the A protein. The SPA allows us to kind of take away, pinpoint where the A protein is so that we can categorize which kind of Staph aureus we actually have. And this is the machine that we use for it, and it's a 3-hour-long procedure, and it's basically just keeps redoing the same thing. And this is the machine. Then we also have a real-time PCR, which we're using for our environmental portion of it. Another thing that we do with this is for the fecal. So real-time PCR detects, counts, and amplifies the E. coli, which is an indicator of fecal contamination in pharmaceutical products. Like here we can kind of see by cycle 16 or like somewhere in cycle 14, we kind of see where we can see where the E. coli is present inside a sample. And then in the future, we are going to continue this. We're going to continue doing the human biome along with the fecal and environmental and clinical portions as well. We're also adding in another component about piercings to figure out kind of like body modifications, piercings, tattoos, what kind of bacteria they have as well. We're in S216s from 8:30 to 2:12, and if you want to talk to Dr. Jimenez, he's at his room in S230 from 12:30 to 2 p.m. Thank you.